Yeah, we'll start the today's program. So, on behalf of the Department of Biotechnology and the Bannari Ministry of Technology, we welcome Dr. N.K. Udaya Prakash from the Wales University of Pallavaram. And the summary of his CV goes like this: he's having more than 20 years of research experience after PhD, and he's having industry 6 years and academics more than 10 years. And he worked internationally in Singapore and USA after a PhD. And the research experience goes over more than 15 years. And he received more than 4 funded projects and around 200 research projects so far he has guided. And he has membered in more than 12 national and international professional bodies and organized several workshops and delivered various national and international invited guest lectures, attended various workshops and conferences, and presented more than this is the old data, more than 150 papers in the research and the conference, and published more than several Indian and national and international papers. And his citations goes more than 1,200, and H index goes around 14 and I index goes around 21. With this few once again, we welcome Dr. N.K. Udaya Prakash for this virtual guest lecture of today's session. Sir, the forum is yours. Over to you.
Thank you, Dr. Kannan. Yeah, my association with Bannari is primarily from the year 2005 when Dr. Kannan has joined the Bannari Amman Institute of Technology. Then related to my personal relationship with Dr. Kannan is concerned, it's more than 25 years we are associated. More or less, he's like a brother for us. We are family friend. And of course, Dr. Arunadava Das and Dr. Suresh Kumar, they're also good friends of me. My juniors, Dr. Vidya and Dr. Vimalarasan is also working over there. So my association with Bannari is not so meager. We have a very strong relationship with Bannari Amman Institute of Technology. Good morning, dear colleagues and my student friends.
Good morning, sir.
Thanks for giving me this opportunity to be with you. Yeah, related to my title is concerned, Aerobiology, where microbiology meets the atmosphere. Actually, I've restricted the title as microbiology, which meets the atmosphere, but aerobiology is not a simple term, it's wide. And it's an interdisciplinary science, which possesses plenty of associated disciplines. Dr. Kannan, thanks for introducing me too. Right, let me get into the subject directly. What is aerobiology? The study about the airborne biological particles. Biological particles in the sense it involves everything. Pollen grains, insect scale, fungi, bacteria, virus, even nanoparticles, enzymes, then other biological oriented materials. When they are suspended into the atmosphere and when you are studying it, then the discipline comes under aerobiology. This is what the general term is. The bioaerosols. You may be knowing when you're sneezing or coughing, you are generating bioaerosol with the droplets. These droplets, along with other biological materials, consist of a general term bioaerosols. What they possess? The bacteria, fungal spores, actinomycetes, pollen, algae, plant cells, insect and mite fragments, their excreta, proteins, enzymes, antibiotics, endotoxins, mycotoxins, everything is there. When you're working with an industry, you can come to know about the biological origin of endotoxin and antibiotics, the laboratory contaminants, even those includes. Right, let's come into the stratification of atmosphere. How the atmosphere is classified into many zones. When you are traveling through flight, then you can, most probably, you might have noticed that in your flight, it may be recording showing that 36,000 feet above sea level. That means we are traveling at the height of 12 kilometers. So we are crossing, nearly reaching the bottom layer of stratosphere. So atmosphere is classified into thermosphere, which is 90 kilometers above altitude of sea level. mesosphere that poses 50 to 85 kilometers. In mesosphere, you can see meteors here flying here and there. The weather balloon, what we are sending to the atmosphere to study about the weather condition, that reaches nearly 40 to 45 kilometers to know about how even the health of ozone looks like. Then few of the jets, they pass through the ozone and have a air flight by 12 kilometers. Then related to your biological origin, that is the materials are concerned, mostly they can fly around our concern is more or less related to 1 meter to 50 meters because the residential height of. So we are thinking about the exposure of us towards the microbe alone. Then the microbiology right here meets you in atmosphere. That is the term aerobiology. Branches. In general, aerobiology is classified into intramural and extramural aerobiology. Again, based on the material what you're going to study, if you're going to read about the pollen grain suspended in air, we call it as aeropollenology. If you're going to read about the fungus spores present in air, we call it as aeromycology. When you're going to study about the bacteria, then we call it as aerobacteriology, and similarly, when you're doing the virus, we call it as the aerovirology. This is what the branches of aerobiology is concerned. Want to introduce a little bit of history. Any subject without history is no use. So just snap on it. In 1850, Perry McHale is the first one to study the bacteria and molds in the atmosphere. That was nearly 170 years before. So aerobiology is young. By 1938, Fred Campbell Mayer, he has given the term as aerobiology. Plenty of work has been done by Professor Gregory from UK, Rothamsted Experimental Station related with studying about airborne pathogens. So P.H. Gregory is rightly termed as father of aerobiology. And this has been continued by Lacy from the year 1995. He started studying about the indoor and outdoor air. This is related to UK than what the Indians are doing. India is concerned, when you are taking back our own Vedas, you can know about the spread of diseases through air. That means the spread of biologicals in air system. This was dated back nearly 1,500 to 500 before Christ years. And biblical literatures also, you can see that the wine preparing grape yachts, vineyard and grape gardens are infected with the fungi. And even Jesus used to curse the people, let the disease spread like that. So that disease is spreading through air. So biblical literature also supports the aerobiology. Then, 500 years before, Lucretius, inhalation of contaminated air which causes the foul air that can be removed using Agnihotra, that is by lighting the fire. You can remove it. In our terms, you are fumigating your laboratory. Right. By the year 1873, this is a recorded evidence where Dr. Cunningham, he served as the jail warden. He has studied the spread of disease within the Kolkata jails using he made an instrument called the aerophonic scope. So it's a man-made instrument to study about the airborne microbes. Then he has published a book as Microscopic Examination of Air. Whom you are seeing in this picture is Professor S.T. Tilak. He is no more. Just a couple of years before or a past year, we have missed him. So he is termed as father of Indian aerobiology. He hails from Pune, and plenty of students he has produced, and dissemination of aerobiology in India was greatly done by this professor. And Professor Sunil Mal Chanda, he belongs to West Bengal. He served as the president of the International Aerobiology Association. Professor A.B. Singh, excellent job he has done. He has written an All India coordinated project where he has brought more than 20 centers from India to study about the aerobiology suspended particles related with the pollen grains and fungal material throughout India. And Professor P.K. Nair is a renowned palynologist, and his contribution is towards the aeropalynology. Plenty of work he has done from Kerala. Professor B.P. Vital, Even you might have seen that your Professor Dr. Kannan may be stating about Professor Vittal whenever he is taking the class. Similar thing happens with me and my juniors because we are closely associated. He is the only man who brought aerobiology to the state of Tamil Nadu. Right now, he is no more. He served as a professor in University of Madras, Center for Advanced Studies in Pollen. So I was trained by him in the field of aerobiology. Right. Don't confine your thoughts related to microbiology alone. Aerobiology is not confined with microbiology. It possesses plenty of interdisciplinary science. A part of it you are learning today is microbiology. Right. Why it is called as interdisciplinary science? It has an impact on human diseases, agriculture, forestry, pollution in air, so pollution related to people, pollution control board needs to apply their mind. Meteorology, so suspended materials may cause the weather change. Defense organization, I'm planning to propose a new term called nano war. That may be the fourth type of war which we may face. Right, outer space. So it speaks about astrobiology even. Animal diseases, when it is going to have an impact on human disease and agriculture, it is damn sure that it is going to have an impact on animals too. Biodeterioration, where it is settling, it may cause harm. For an example, archaeological importance sculptures and buildings, they can easily be contaminated, deteriorated by these organisms whenever they are settling onto that surface. Locust movement, you may be heard about locust in Gujarat, Haryana, Punjab, those areas, maybe 3 or 4 months before. Palynology, general ecology and phytogeography. Even you can say the phytogeography. For an example, you can have, if you're getting deposit of finest pollen, you can easily say that it is a hilly zone. Like that, you can even find out the palyno or phytogeography of different locations. So thus, aerobiology is an interdisciplinary science. Right. Here, we can think about the aerobiological pathway, how it is being done. Just by 5 steps, you can calculate the pathway of aerobiology. Source, first thing, if you want anything to be suspended in air, that has to be present at the ground level or from some surface. This is called as source. They release, whenever the air is blowing or wind is blowing, the material needs to be released to the atmosphere. When it is being released, the released content needs to have the flying capacity. So the shape needs to support. Their size needs to support. Shape and size is important. Then other climatic factors will also play a role in their flight. Right. Deposit. How they are being deposited. Depending upon the deposit, that when the wind blows, they can deposit on any zone. Depending upon the depositing surface, it has an impact. So each and every area, environmental factors like temperature, moisture, humidity, rain, everything plays a role. If it is depositing on the leaf surface, if the conditions are favorable, they start growing and can cause a potential disease in the plant. If the same spore, if I am going to inhale it, if it is going to settle on my lungs, then it has a potentiality to cause the respiratory disease. Like that, the impact differs. Here you are seeing how the dispersal of spores and pollen grains are taking place. Just it's a diagrammatic representation. You can see how it is being carried. Then, even in your refrigerator or in your own house, you can have all these infections. Tomato, onion, chili, lemon, everything is getting infected with molds. Even bacteria, the rotting stage of whatever the food material we are having, that is because of air contaminant. Of course, there is no word for me to say about your laboratory contamination. You may be far experienced related to your laboratory contamination is concerned. That is all because of aerobiology, that is airborne spores. Right. Why we need to study about the microbial pollutants? First thing, they are typically acting as a threshold, in the sense they serve as a pathogen, a toxogen, and allergen. This is the unique property of microbes. If you are taking the pollen grain, they serve only as allergen, not as toxin or pathogen. Whereas microbe is concerned, they will be acting as pathogen, toxin, and allergen. So this is the three-dimensional attack where you are going to face. Right. If the size is between 1 micrometer to 50 micrometer in size, they have a potency to reach your upper respiratory system as well as your lower respiratory system. So they have the plenty of spaces for you for them to settle within your system. Source availability. If the source is not there, the spore is not going to be there. Then the buoyancy of the capacity to float in the air, how they are good. So depending upon that, then apart from that, they need to be present in significant number. So if all these things are there, the microbe is going to serve as a potential pollutant where it can easily attack you or any plants or animals. How you can sample? Yeah, right here, just I want to say about the background about the Marina Labs. The biggest laboratory in the field of aerobiology is Marina Labs in India. We have all sort of samplers which are exhibited right here. So that's what I said. We are the richest laboratory, the biggest laboratory in India related with aerobiology. The first one, what you are seeing is liquid impinger. Using that, just you can take a buffer on this. Even you can use the buffer or any other culture media into that. Or this, if you are ordering it right here, it may cost you around 3,500 rupees. Whereas this one is imported with the calibration, so it cost me around 12,000. So liquid impinger is one of the sampler. Second one, Anderson sampler. This is a highly sophisticated. Well, the cost differs from 2 lakhs to 5 lakhs. First one, what we are saying is within Anderson sampler, single stage. That is, you can able to expose a single petri dish. Then the same way, next diagram which shows 6 stage and then the 2 stage. In 2 stage, your lung system is classified into upper respiratory system and lower respiratory system. Then, towards here, the 6 stages one, maybe by next slide, I can explain you about that. Then the next one, RCS, Reutal Centrifugal Sampler. We have imported this from Germany. And this particular sampler, most of the laboratory, biological laboratories have this. But this was made mandatory for the microbiology department in many colleges. But I don't know what the people are doing using the sampler, whether they are working or not. Right. Biosys is the other type of sampler. Here directly you can expose the slide, take the slide back to the microscope, you can view it. So the reporting time is reduced nearly to only 3 hours. Whereas when you are depending upon the culture, it may take 2 days to 7 days. So that can be reduced, particularly when you're serving in industry, you may be knowing the significance of the time. So whether what the contaminant and what the level of contamination occurring when the production is going on. So this helps you a lot. The same biosys sample is modified into aerosol, that is use and throw, made up of plastic. This is used for a single usage. After usage, you can just throw them. Then comes the cyclics and biosys. There is not much difference. Only thing, their orifice is different. In cyclics, it is circular orifice. In biosys, it is slit-like opening. Burkhardt personal sampler. Here again, the opening is slit. You can use the slide, coated with the Vaseline. After exposure, you can take the slide, take it directly to the microscope. You can view then what are the types or what are the quantity of fungal spores or microbes present. You can easily record. Right. As I said, the 6-stage Anderson sampler here, it is a diagrammatic representation showing that where the microbe can deposit in your upper lung, in your respiratory system. Right here, stage 1, that you can have the microbes which are 7 microns and above. So they have the capacity to reach your upper respiratory system. Stage 2, 4.7 micrometer to 7 micrometers. This will reach your pharynx zone. Then stage 3, trachea and primary bronchi. Stage 4, secondary bronchi. Stage 5, terminal bronchi. Stage 6, your alveolar zone. If the microbe is reaching your alveolar zone, Your body temperature is 37. It's a cozy temperature. Moisture is going to be there, and it is encircled, right? Even if you want the nutrition, it can be supplied through the blood capillaries. Just they'll start licking, and then they feel cozy. They start growing, occupying your lung. So if it is going to occupy your lung, and if it is going on a, your respiratory rate and respiratory volume get reduced. So that results in the disease called as ABPA, obstructive bronchopulmonary disease, ABPA, right? Or obstructive bronchopulmonary aspergillosis. Here you can think about the wonderful fungi Aspergillus fumigatus. That particular fungal species have an ability to grow from 20 degrees to 55 degrees. If you are taking the mean value of the temperature, it clearly falls under 37 degrees. So your lung or your human system is going to serve as a beautiful area for the fungus to grow. Then the microbes, the spore size is just less than 3 micrometer. So they have the capacity to reach your state easily, right? So have that in mind, how the microbes can cause disease, specifically related with the respiratory one. Right. Okay, so sampling. Frankly speaking, 1 hour time is not sufficient for us to discuss about these things. I have capsulated into a small capsule and trying to present it for you. Now, let us know about the intramural aerobiology and the extramural aerobiology. Intramural in the sense, what is that intramural aerobiology? When you are going to study about the presence of microbes within the confined environment, that is, having walls on all 4 sides, covered with a terrace, then you call it as intramural environment. Extramural in the sense, they don't have any close, wall structure at all. It is completely open. So the list of things we have listed on your left all belongs to intramural. Home, intramural. Hospitals, hotel, industries, places of worship, educational institutions, animal houses, transport system, malls, everything falls under intramural one, right? So even you can add more things to this. Extramural in the sense, garbage dumping site, market, highway, agriculture field, monitoring the entire city. For your information, there is no report available from Erode so far. In the state of Tamil Nadu is concerned, only Chennai is widely studied. No other city is studied. However, Dr. David from Karunya University, he has studied about the Coimbatore Corporation. Trichy Corporation, I have completed. Vellore Corporation, I have completed. Or Trichy, Vellore, Chennai alone, we have completed on our own. So there is plenty of opportunity for you people. Whatever the report you are going to report from Erode, that is going to be the first report and acceptance is easy. That is going to be the first report for the state of Tamil Nadu. Keep that in mind. Right. Public environments. All this falls under extramural aerobiology. Let us travel a little bit into extramural environments. The roadside. So few of my BTech students, they have done their project related to the microbes present in the traffic signals of Chennai city. So here you are seeing the carpet chase sampler, then the gas sampling pump connected with the current system. Of course, here Dr. Kannan also participated in this study. We have operated our sample in Marina. So we want to know about the study related to the airborne fungal and bacterial count in the coastal zone. We have studied this from Mamallapuram coast to Yennur coast. We have studied nearly 17 different sites, covering around the 5 meter, or 50 meter, and 100 meter distance away from the coast. So yet to be published, but we have shared it in some conferences. Right. Yeah, this is what you are seeing it in Mamallapuram. This is a beautiful zone. The farmhouse, just see the amount of dung material present there. 1 ton illa 2 ton illa kittathetta 5 ton of dung material available in a single site. Then think about the people staying in this area. Continuously they are exposed to the methane, second, the fungal spores, bacterial spores, other volatile organic compounds produced by the microbes. Continuous exposure is there. Think about their health system. This is the other view where our samples are working, and you can see the people who are working in that as well as the residences on at the last of this particular photograph. So you can see the kids always with the, having the running nose here and there. That is because of their exposure to the microbe, then imparting allergy, resulting in running nose, right? So their occupational exposure is heavy. Then this is their health is also compromised because to provide the milk for you people. This is a beautiful site where you can see the cattle are lying on left and right, and then on the middle we are taking the sample. You can see the dung materials apart from the urinals, and then the hair material they will serve as the moisture supporting as well as other support to this particular microbial growth in this particular environment. Other area it is vegetable market. So you may be knowing the infection caused in vegetables, might have seen, and you might have thought that. Think about the presence of microbes in vegetable market, plenty in count. Then think about the people who are working in there, just vegetable vendors. They are continuously exposed. If I'm asking you to visit the vegetable market and procure few vegetables, then what you say is, no, we'll feel nasty. Like that we are reporting. But the people who are working over there nearly for 24 hours, they are continuously exposed to microbes. Keep that in mind. These are all extramural environment. That is, they don't have any socials with them. Right. So the other view of vegetable markets, here what you are seeing is meat market or fish market. Our study, we studied around 25 different fish markets in Chennai. Particularly, we concentrated on the presence of bacteria. For your shock, we isolated Vibrio cholerae as the dominant one in these areas. Even I never expected that. Actually, we thought Micrococcus may be dominating, but the result showed it is Vibrio cholerae, which is dominant in these environments. So think about the exposure of all these people who are serving in that particular environment, fish market. Then what about their health? You might have even experienced the report stating that North Madras, but adjacent to Kasimedu, frequently reporting cholera disease. So this is possible. Now you may be knowing the reason. What is the reason behind it? Here we are exposing our impinger to collect the airborne biologicals, later taking it to the laboratory. We'll be examining it for the presence of bacteria as well as virus, then classifying them according to the genus. Finally, we can make it as a report about the bacterial microbial presence in fish markets. This is again an extramural environment. Solid waste dumping site. Even a small can of rubbish next to your house, you feel stinky and reporting it. Why is this happening? Because of putrefaction of all the material. See, this is a beautiful substrate which possesses cellulose, fat material, as well as plenty of proteins. These are all denatured by the microbes. So lot of microbes will be present in this particular environment. So whenever some disturbance is happening or a wind blows, they used to come into the atmosphere. So when you're working in solid waste dumping site, you're continuously exposed to this. For your information, a personal discussion with the people who are serving in this particular solid waste dumping site, they used to report tuberculosis as their disease at the age of 55. So they used to die at the age of 57. In India, the longevity age is 65. They are losing nearly 7 to 8 years of life just to keep you clean. Keep that in mind. Just to keep you clean, they are losing their life for nearly 7 to 8 years. But we are least bothered about their health. Just we'll be throwing our rubbish here and there. So now you may be knowing the significance of Swachh Bharat, which has been brought. So keep that in mind, maintain as much as possible about that. Right. The other view in solid waste dumping site, Dr. Kannan may be having the personal experience because he used to accompany me whenever I'm going for sampling. So he has also experienced about this particular nature of solid phase ducting site. See the beautiful site. Think about the lorry driver. Think about the cleaner. Then the people who are just bringing about the trashes of rubbish. Why we are not thinking about their health? So apply your mind, the aerobiology that makes you to think about the health of others. That is the beauty of this particular science. Right. So let us get into the intramural environment. Intramural environment is concerned, they are enclosed with wall. So it is an enclosed environment. Food preparing unit. My student, Dr. Bannari, she has done the work related with the airborne microbes in food industries. There's just a small bakery where they are preparing the food. She's serving as the professor in MGR Janaki College. Well, see all these cute pigs, guinea pigs. Whenever you're going near them, they'll be screaming, "Pack, pack, pack, pack," like that. That's beauty. But when you are entering into that, the smell emanating in that particular area is intolerable because of the urinals, the shit material, that is the dung, the moisture. Then they need AC. Only by 22 degrees they can survive. They want AC, so they feel cold. Right. So that particular environment serves as an excellent excellent microclimatic condition, provides microclimatic condition for the microbial growth. The rabbit house, similar condition. Can anybody see what is this animal? When I'm directly asking, then many of the people used to say this as the rabbit, or they used to call it as tiger. Right option lies with you. You think on your own. Later on, you can even give me the answer. So far, I have not ventured the work related to these animals, how their cages will be. And here you are seeing my students who worked on the project related to the microbial exposure of zoo captivated animals. The one behind, she is serving as a collector in Tiruvarur district right now. She has done her BVSc, Veterinary Science, in Madras Veterinary College. Again, food preparing unit. These are all the enclosed environment. Factory, you can see the dove which is coming into the air. Think about the man who is working there. Even he's not having any mask. Then think about his respiratory level, how much he might have inhaled the dove material getting into the lungs. Do you think that it may get digested? Think about his health. Yeah, we have served as a consultant for one of the biggest hospital in Karnataka. As we need to maintain the secrecy, we are not revealing the name of that particular hospital. So they want us to monitor the microbial level in ICU. Why it is called as ICU? You are not allowed within. So you can say ICU. So it is rightly termed as ICU. So we have studied about the microbial presence in ICU of one of the biggest hospital in Karnataka. We have made a report, and we have also done the same study for Nokia and for Apollo hospitals. So studying about the indoor air quality is important, one of the branches of aerobiology. Here you can see the toilet even. Here I want to bring the attention of you people. The best job for the biotechnologist or the microbiologist is housekeeping, because only we know the quality of microbe in any environment. So if you are taking this particular job, it is completely professional, right? So don't miss this opportunity. Plenty of corporate officers are there. Even we can take the tender. We say that we will be monitoring it. Plenty of money we can make this offer, right? One of the business. Keep that in mind. Can you guess what it is? நீங்க செத்து விளையாடி இருந்தா இது எந்த ஏரியான்னு தெரிஞ்சுக்கணும். ஜனவரி பிப்ரவரிக்கு அடுத்தது இதுதான் வரும். நம்மளுக்கு இந்த வாட்டி கட்டாயம் வந்தது because of corona. By March, this is Marchvary. So my students they worked on the microbial presence in mortuaries. This is one of the biggest hospital again in Pondicherry. Right. So you may be knowing, by this time you may be knowing what are all the areas you can study, what is intramural and what is extramural, and how they used to differ. There is plenty of area for you to do research because there was not much research in the state of Tamil Nadu, except myself and my wife. Most of the people are not working related with airborne microbes. So there is plenty of opportunity for the people to study. Right. Here just I am giving you the pictures of the rest one. See how the microbial growth is. Then the third picture, what you are seeing is whenever you are playing to maybe in your younger age, if you younger age, maybe you might have reached older, hide and seek. You might have hide in those areas, then exposing to the microbe. Finally, you will be saying and get caught. So hereafter, when you are playing hide and seek, first seek, then hide. The samples what you have taken, how they look like in the microscope. The beautiful ketomium. See how the spores look like. Beautiful ice-like, right? You need, let's say, an appreciative mind. Look into the microbe, right? Nigrospora, typically like your bindi. Plenty of Alternaria spores. Aspergillus. This is the biofertilizer trichoderma. Then trichoderma along with the membraniella and then the rhizome. People in US are scary about this, Stachybotrys. They used to call it as toxic mold. So this is the Stachybotrys. Again, Eulocladium, Cladosporium. A cocktail of all these things, Stachybotrys, Alternaria, Cladosporium, Aspergillus, plenty of spores. And this is the beautiful spore what we have recorded from the atmosphere in US. So whatever running like a snake, they are termed the Cercospora. By this time you might have called the disease caused in Arachis-hypogaea. Cercospora arachnidicola and Cercospora personata, causing the Tika disease of groundnut. So whomever is coming from the background of agriculture, they may be knowing about. So this is a beautiful fungal spores. Then you can see Alternaria, Cladosporium bunches. Again, it's a beautiful stuff for any aerobiologist to have a view on this. Here you are seeing plenty of different insect wings. This I have taken it from the shit sample of the bat in Austin, Texas. This is the world's biggest urban bat colony. So you can see plenty of different type of insect wings. And again, a cocktail of microbes. Here you can see Cavalaria, Oidium, Rixlira, Fusarium, Cladosporium, smut spores, Periconia, Fusarium, Cercospora again. Again, it's a beautiful cocktail. Just you guess, this is just your skin material, your own human skin. The dried human skin looks like this when you are staining it with lactophenol cotton wool and experiencing it under the microscope. Culturable molds, most probably Dr. Khan might have taught you about plenty of culturable molds. Particularly as an endophyte, you might have experienced a lot of culturable molds. Here I'm giving just a few samples. Even as a contaminant, you might have experienced a lot. The beautiful petri dish, as a palette, it is looking like a closer view of Cladosporium. You need again a mycologist's eye to appreciate this. Penicillium producing different exudates, typically looking like an omelette, the colonies of Ketomium. Right. So you say that you are the biggest laboratory in India, and then இதெல்லாம் claim பண்றதுக்கு ஒரு authority வேணும். What the hell you have done for Aerobiology? இது உங்களுக்கு கேட்டிருக்கலாம் மனசுல எழுந்த question. For any people who are working in science, அவங்களுக்கு ஒவ்வொருத்தருக்குள்ளேயுமே இந்த கேள்வி இருக்கும். So same way for me in the field of aerobiology, I'm not supposed to justify why I am in this field and what my contribution to this particular field is. Right. We are the first one to report Chennai pollen calendar. This is what the working group is. This was supported by Dr. Raju. During those days, he has supported with 1,30,000 money for us to study. You can see, for an example, if I am taking the month of March, I can have the plenty of bars running, which shows in the month of March, you can record only those pollens at what level. Next to the total pollens recorded, the key is given. So when I'm circulating this to any allergy specialist, they can easily say by the month of July, okay, these are all the allergens, pollen allergens present in the atmosphere. So they can treat the patient only for that. So such a huge contribution is this. This particular Chennai pollen calendar was released by the former governor of the state of Tamil Nadu. In the middle, you are seeing Professor Dr. A.B. Singh, who served as the secretary of both Indian College of Asthma, Allergy and Applied Immunology and Indian Aerobiology Society. And then Dr. Udaya Prakash, who is receiving the copy from the governor. So this is what our contribution related with palynology is concerned in the state of Tamil Nadu. Then we are the first one to develop unmanned aerial vehicle, UAV, to monitor the high altitude spores. So we have designed this particular thing along with WellTech. Of course, I served as a former professor in WellTech. I had an excellent team. Dr. Vasantaraj, Dr. Balasubramaniam, and then few of my students. We started working with designing this particular unmanned aerial vehicle. For your shock, it is working with the battery. Previously reported from different countries, they used to have the fuels. Here, we use battery alone. So environmental friendly, carrying the sample on both the wings of flight, then it captured the fungus spores, we cultured. Finally, we have published it as a paper. Right, so here when we use this particular sample, we felt that the height, weight of the sample is imbalance in the flight. So we thought of designing a new sampler which is less in weight. So this weighs only 35 grams. This is what we have designed with the self-propelling system. So on its own, it starts propelling, sucks the air, and then we used to get that filter. We study the presence of bacteria as well as fungi. This is actually here, don't copy this particular stuff because under pattern, this is what the multi-utility air sample we have designed. If you are using this in a single shot, you can isolate actinomycetes, bacteria, fungi together. Apart from that, if you are concerning about, you can use different type of media even. So nutrient agar, potato agar, or even algal agar, everything. Dr. Kannan, I don't have the video right now, so it is difficult for me to play about the UAV, I think. Maybe by in person I will send. You can share it with the students later. Right. Then we felt the fixed wing UAV is having not having the hovering capacity. So we have designed the multi-rotor system. Even now we are working with MIT, Chennai, to bring about an excellent system for monitoring the atmosphere. Here you can see our sampler fixed with a multi-utility rotor, or you can call it a squad rotor. So this is again an interdisciplinary science where we are taking the help from mechanical engineering. You can see that our squad is flying at the height of 1.5 meter altitude. Even we fly this above 100 meters, then we started monitoring. The report yet to be done. We need to publish the paper yet. Right? So towards its application, in agriculture, you can forecast the disease. That is the beauty of aerobiology. So by the day 1, you are getting 20 spores. Day 3 or day 4, 50 spores. Day 7, 130 spores. That shows the particular pathogen has started multiplying. So it is going to affect all other field. By that time, you can just inform the farmer stating your plants are going to be affected by this disease. So start spraying the fungicide or bactericide or viral related pesticides so they can save their crops. So disease forecasting is possible. Veterinary science, studying about the epidemiology of animals. Medicine, toxicology, again epidemiology, allergy, dermatology, everything. Pharmaceutical science related with quality control. Just think about the pharmaceutical product. If it is going to contaminate it with your microbe, no use. So here comes the beauty of the aerobiology. In hospitals, quality control, indoor air quality, occupational therapy, then even the cases of ICU. I want to give a case. Former minister got admitted with a very big hospital in Chennai. He underwent bypass surgery. After 2 years, again he was admitted, but he died. The reason said by the hospital, what they said is, your pacemaker got infected with fungi. What? Then how about the environment of operation theater? Your pacemaker got infected with fungi. Because of that, the person died. Former central minister. Keep that in mind. Right. Architecture in hotel. When you're staying in room, next day you'll be reporting allergy, particularly with AC rooms. Your kids may be reporting fever or allergy. That is because of the microbial quality of that particular hotel room. So that has to be looked into and certified. Care of microbe-free. Architecture, indoor air quality, building related illness, unsick building syndrome, vastu sastram, எந்த ரூம்ல இருந்தா disease வராது. So it's an indirect way of aerobiology. Food industry, quality control, environmental monitoring, then biotechnology, that is the biodiversity. BPO, now it has also grown into BPO, where you can do the service. Just collect the slide, examine it, give the report through email. So such a huge applications are available. I've listed very few. Even if you are applying your mind, you can add the list to this, right? Centers of aerobiology in India. What are all the centers? Professor Yebi Singh, Bhattagar and Gaur, they have worked in Delhi. Professor Yebi Singh got retired. Professor S.T. Tilak from Pune, called as father of aerobiology. He's no more now, but his students are carrying few works. Vittal and Udaya Prakash, Vittal, the doyen in aerobiology in the state of Tamil Nadu. Now we are continuing, myself and my wife, Dr. Bhuvaneshwari, we are continuing with the research related to aerobiology. N.I. Singh from Impal, he got retired. Even he said, if few of you are interested, he is ready to share his research experiences, that is the papers he published and the collections what we have. Then Sri Ramalu, no more. Subareddy, no more. Dr. Janaki Bai got retired. Dr. Ramachandra Rao, he is a beautiful man who worked with the airborne algae. Right now he is serving in Gulf countries. He is the man who gave me the clue to relate, to work with the nanomaterials suspended in air. So right now I moved on to the nanomaterials suspended in the atmosphere. Then Dr. Jogdhan and Dr. T.N. Murray, they worked on the mite. Dr. B. Rangoswamy, he is again a type of specialist. Melitopellinology is his specialty area. He used to study the pollen grains of honey bees, honey traps, everything. Maybe Dr. Kannan can fix a talk with Dr. B. Rangaswamy of Davangere also. That may be of interest to the students. Dr. Savaji from Nagpur, Dr. Tiwari from Raipur, Dr. Pandey, Aurangabad, Dr. Tirike, Amravati, Dr. Karuna Verma, Jorat, Dr. Chanda, no more. Dr. Kashinath Bhattacharya and his wife is taking excellent job in Shantiniketan, that is in West Bengal. Dr. Agashe got retired. Dr. Manjunath is studying about the few aerobiological things in Bangalore. Dr. Vikat Jain from Bekaner, Dr. Manju Sahani from Allahabad. So most of the faculties got retired. Now, plenty of lacuna is there for the youngsters like you to take the area, right? Right. We are offering a free online diploma course in aerobiology. Already the course is on. Nearly 46 students have registered, and currently the course is going, but the assignments yet to be given. This is what the form nature, where you are supposed to fill. If you are interested, you can also fill this form and mail us, and you can join the course. It is open right now. What it possesses, the syllabus. I have shared all these things with Dr. Kannan. If you are interested, you can reach him personally, can get the details related to. Then this is second paper, theory. And practicals, I'm wondering whether I can able to make it or I need to, because of this COVID situation. Then certificate will be provided, but we used to charge 1,000 rupees for certificate because this is a full proof certificate where duplication is not possible. So we are charging 1,000 if the students are interested to get the diploma certificate, or else it is completely open for everyone. It is not necessary for you to be the biotechnologist or biologist. Anybody can join, even the chemist can join this particular course. Right. Towards the association, when you want to become as a member of Aerobiology Association, Indian Aerobiological Society is there. In Italy, Associazione Italiana di Aerobiologia. In Norway, Nordic Aerobiological Federation. France, Society Francaise di Aerobiology. US, Canada, and Mexico, Pan American Aerobiology Association. UK, British Aerobiology Federation. Then Switzerland, Schweizerisch Gesellschaft Aerobiology. Everything put together, you can have International Aerobiology Association. So these are all the association, or you can become as a member of all these associations, right? This is what the journal, Indian Journal of Aerobiology, is brought by Indian Aerobiological Society. Then we have shared our course in this YouTube. If you want more details, you can also, about the Marina Labs, you can also follow us in Facebook page or https://www.facebook.com/RNG Marina Labs. There you can follow us. So even about the course details you can get. Then by Tuesday, I'm sharing the experience of or the perspective of aerobiology just related with COVID-19 with youngest University of Madras, which is serving for Ministry of Forest, Environment and Climate Change. That talk is by Tuesday. I'll be sharing the, even you can follow us in Facebook, get the details about it. Or when you are registering, you can get the certificate in that particular course, which is given by the University of Madras. So that there I'm going to speak about the aerobiologist perspective related with COVID-19 in India, right? Yeah, if you have any queries, you're always welcome. Yeah, if you have any queries, you can ask.
Thank you for your the overview about aerobiology and your personal experience in this particular field. And now I request the audience, particularly the students, if you have any questions in your mind, you can feel free to ask. Now the forum is for discussion.
I need to thank Dr. Khanna, the organizing secretary, the principal of the institution, the R&D director, and then all of the faculties for patient listening, and students. Thank you for your listening here. Right. Yeah, your queries are welcome.
If you have any mic issues, you can chat, you can put in the question in the chat box. So if you have any mic issues, don't worry, you can post the question in the chat box. Yeah, please. Hello.
Yes.
Sir, good morning, sir.
Right. Yeah.
Yes, sir. Sir, long back, I think 5 years ago, I got meet you in the Science Inspired Camp, sir.
Oh, thank you. In which institute?
Sir, IFET College of Engineering and Technology.
Oh, right, in Vilpuram.
Yes, sir.
Okay. Thank you.
Glad to see you again, sir, and it's a very nice session, sir. Thank you, sir.
Yes, glad to see that you are in Bannari. Great, man.
Yes,
when
sir.
I'm coming there, I'll meet you personally.
Okay, sir. Thank you, sir.
Yeah.
I was waiting to see you, sir.
Thank you.
Yeah, Tillayarasan, thank you. And there is a question in the chat box.
Sivaranjani, what
Sivaranjani.
initiative can a common individual take to keep the air around the skin safe? I'm also thinking on that angle. You are the one who needs to support me. You can think over that. You can come out. Just think about how they are controlling the COVID. Few people are working with UV, fumigation, if you want me to say in our own cultural system, avans, yago naduthuvam. So that will replace. So fumigation is the best one. This is what even hospitals are following in the operation theater as well as in ICU, right? Sivaranjani, you got cleared with your question? You can type even. Thank you. Right, any other questions?
Any more questions?
If it is not there, if you convey my thanks to all the organizers and the student participants, and then to Vidya, Vimalarasan, Dr. Suresh Kumar, Arunavadas, and
Vidya
Dr.
ma'am
Vidya ma'am is there. Okay.
So, if there is no more question, so I thank once again the Dr. Udaya Prakash for their ready acceptance for the virtual guest lecture, and on behalf of the Department of Biotechnology and the Bannari Ministry of Technology, on behalf of the staff and the students, once again we thank you for your valuable time. So personally, we thank you once again. So thank you.
Kannan, Dr. Kannan, shall I find out?
Yeah, you can please.
Yeah, thank you. Bye-bye.
Yeah, thank you. I will share the recording slow.